Hello, I'm a little bit, it's a bit strange because I can just see my own talk. But anyway, I hope there's people watching me. I'll just see if I can get my second screen up and try and, yes, I can. Just 1 minute. Okay. So I can see what you're seeing now. Oh, okay. So I would like to talk about apple trees and DNA fingerprinting. So my name's Mark Winfield, and I'm from the University of Bristol, and I work in a group of people who mainly work on wheat, but I'll explain why we got involved in this work as we go along. So this picture here is of the National Fruit Collection at Brogdale, where we went to 2 years ago and collected a lot of samples, but I'll talk about that in a moment. So let's get there. So the outline of the talk then, the first part, and this fits in really well with the keynote speaker this morning when we're talking cider and women, because I want to talk about research that took place at Long Ashton Research Station, and it produced a collection of cider apples, of cider apple trees, collectively known as the girls. So I'm going to do a little bit of introduction to that. Then there'll be a section a little bit more technical in which I'll talk about fingerprinting and how it works. And in order to do that, I'm going to have to talk a little bit about DNA, so I'll show what that is, and what SNPs are. Then I'll talk about something that we call minimal markers, so it's a small number of SNPs that allow you to identify varieties, and we use that on the National Fruit Collection at Brogdale. And finally, I'll talk about the apple hunt. The first round of the apple hunt took place just before lockdown happened, and we got the results at the beginning of lockdown. And now we're just about to start a second apple hunt, and would like to invite people to offer some of their apples from some of their orchards for inclusion in this study. Okay, Long Ashton Research Station. The research station itself had its origins in 1893. The experiments that took place on a farm in Glastonbury between 1893 and 1903. And at the end of these, the people who were doing these experiments decided it would be a good idea if they could develop an institute for the study and improvement of West Country, the cider industry. And that ended up with the establishment of the National Fruit and Cider Institute. It was sited on land belonging to Ashton Court Estate, which is just the southwest of Bristol, which is most famous now for its ballooning days in the early summer. But anyway, in 1912, that institute was taken over by the University of Bristol and became the Department of Agriculture and Horticultural Research, and became known as Long Ashton Research Station. And over the years, particularly in its early years, it gained a worldwide reputation for its work on fruit and cider. And as I said, it fits in really well with the idea of cider women, because there were a lot of women involved in the cider making process and working in the orchards. So in the early years, particularly, I think Long Ashton was responsible for a resurgence in interest in pear trees and in perry making, and in some ways helped to revive that industry. But obviously, we're more interested in the fact that it was also involved in the cider industry. It's perhaps best well known for a different project. So in its early years, in the 30s, in 1933, Dr. Vernon Charley was doing studies into the health-giving properties of vitamins and fruit juices, and particularly vitamin C. And he was trying to develop fruit syrups that could go into milkshake manufacture using pectinases as a part of that process. Well, it didn't have much success as a milkshake, but it had a great deal of success as a cordial and became named in 1938 Ribena. And because of its supposed health-giving factors, during the Second World War, this cordial, without its name, was distributed free to children and pregnant women. And if you look at the advert in the middle, they make some quite exaggerated claims about it. In later years, in more recent years, this Ribena has fallen somewhat into disgrace because of the high content of sugar it contains and apparently the low level of vitamin C. But let's move on from that. Let's get back to apples. So long-action research then, in later years, so this is now coming to the 60s, started doing work with cobalt-60 irradiation of samples to try and create spontaneous mutation. And they did this with some scions from Bramley seedling trees and produced the Bramley clone 20, which is a mutation of that. The Bramley seedling tree is quite a large, vigorous tree that is probably too big to grow in most people's gardens. The wonderful thing about the Bramley clone 20 is it's a smaller tree, about 20 cents smaller, but still bears fruit, large fruit. The cobalt-60 experiments also resulted in the production of the self-fertile cocks. And now we're talking about Bramley, we were lucky enough to go up to Nottingham and see the original tree, which is in Sutherland, Nottingham. It's now a very sickly tree. The original tree is dying. It's got honey fungus infection, and it's now owned by the Nottingham Trent University. The house is as a student's accommodation, but by doing that, then they can look after the tree to some extent. Now, in the 90s, Professor Cocking from the University of Nottingham, and I did my degree at University of Nottingham, or was awarded it from Nottingham, although I did work my PhD at Long Ashton Research Station. Anyway, Professor Cocking, in his attempts to save the tree, did some clonal propagation of that and produced clones from the original tree. Anyway, let's get to the cider apples in Long Ashton. So late 70s, early 80s, I believe. And again, this brings us back to some of the women in cider or cider women. Some of this work was done by Liz Copas, who was mentioned this morning. And there were some crosses set up between cider apples, Dabinet and Michelin, and dessert apples, James Grieve and Worcester Pomaine. So the cider apples were the females in the cross, and the dessert apples were the males in the cross. And the crosses were made in all combinations, so Dabinet with James Greaves, Dabinet with Worcester Pomaine, etc., etc. And I think the outcome of this was something like 1,500 seedlings. Now, over the years, these were selected to see which were the best, which had actually useful qualities. And I think as late, it was only in 2007 when finally a selection of 29 new trees were made. And the vast majority of these had taken on names of women who were working at Long Ashton Research Station in the pomology department. And these are some of the names of the female trees that were produced from that cross. And I've just got a nice picture that shows some of the women working under the apple blossom there. So there were these 29 trees, they're collectively known as the girls. Unfortunately, Long Ashton Research Station closed in 2003. It was sold by the university. Now, that picture on the left is the laboratory in which I did my PhD. I have to admit my PhD wasn't on apples, but it was on wheat, because in the 80s, the institute changed its focus due to changes in funding, from working principally on fruit to working on cereal crops, and my PhD was on wheat. Anyway, the land was sold. became a housing estate. And as you can see, they also established a day nursery there, and it took on the name of Apple Tree, so that even the data reflects the fact that there was apple research taking place at the station. Anyway, now let's get to the part about the DNA fingerprinting and analysis. So the cider apple collection moved from Long Ashton to the university's Goldney Gardens, and obviously samples were taken to Thatcher's and other orchards as well. But unfortunately, in the closing, which was its centre year, by the way, in 2003, in the closing of the institute, some of the records of the crosses made to produce the girls, these were lost. So people from Long Ashton, Les Copas, came to other people at the university who were involved in genotyping, but people who had actually worked at Long Ashton, so like myself, Gary Barker and Professor Keith Edwards, and asked if we could help to identify the parents of the girls. So, as a group, we're involved in high throughput genotyping in wheat, which is a little strange. We should be doing any work on apples. So for the last 10 years, we've been developing high throughput genotyping arrays with up to 800,000 SNPs and 800,000 markers on an individual array. And we've been doing analysis of an extensive range of wheat varieties. And I actually maintain a website that makes all that data freely available, and that's called CerialsDB. Now, just to show, as I came to this meeting, I thought, well, I'll be a little bit of an outsider, but it's not been like that at all. And even from the very first talk by Andrew Lee, there was some connection. And I was feeling rather embarrassed because he said that he'd gone to Kazakhstan, into the Tian Shan Mountains to look at apples. Well, probably at the same time, it might have been a year later, I went there with a group of wheat geneticists to look at the wheat lines, the foothills of those very same mountains. So at Almaty, the father of the apple. And just by coincidence, it just happens that Kazakhstan has some land races of wheat that contain a lot of genetic diversity, which would be useful in wheat breeding. So anyway, let's get on to the story now. This is where I want to talk a little bit about DNA and what DNA fingerprinting is. So this is a nice diagram that I found on the internet. I've not done it myself. The pictures on the left are mine, though. So we all know that organisms, multicellular organisms, are made up of individual cells. So you've got the cell with its nucleus, and you've got a picture on the left is of cells, of wheat cell protoplasts. So this is the work. This is me going back to my work I did with Professor Cocking from the University of Nottingham, is producing protoplasts from individual cells. You use enzymes, pectinases, heliocellulases, cellulases to digest away the cell wall. So these are protoplasts that I created in the lab for some experiments we're doing, and that's what they look like. They have a nucleus, which is where the DNA is. We can't see that in those photographs I've taken on the left, but if you use special techniques, you can. So this is using fluorescence microscopy, and we've introduced into these cells a fluorescent dye which is targeted to the nucleus, so we can see the nuclei in there. But this is important just to make out that you can introduce colorants that you can see with a microscope, and we'll come to this later on when we're talking about SNPs. Okay, so we can see a nucleus. Within the nucleus, above that, we have a picture. Again, these are wheat chromosomes of chromosomes, and you can see they more or less have the appearance as in the drawing. And we know that the chromosomes are made up of DNA wound around proteins. But if you unstretch all that, you can see the individual strand of DNA. And DNA is just made up of these 4 different nucleotides: guanine, cytosine, adenosine, and thymine. Which means in the end, And from a molecular biology point of view, you can view DNA as a simple string. And this is a real string from apple. I pulled it down from some of the apple genotyping we've done. This is just a piece of DNA, and it can be represented as a long string of letters, A, T, C, and G. There might be a few other letters in there, but they stand for ambiguities when you weren't quite sure was there. In apple, there are about 750 million, so 750 megabases of DNA in a single nucleus. So the apple chromosomes. Apples tend to normally have are diploid. They have 17 chromosomes in copies. So just to look at the human genome, which you'll probably be more familiar with, humans have 46 chromosomes. They tend to be in pairs. So we are diploid organisms. Most apples, probably 80% of apples, are also diploid. So the chromosome, apples have 17 pairs, humans have 22 pairs, and then the X and Y chromosome. When they're talking about the amount of DNA in your organisms, the 750 megabases refers to the haploid complement. That means what would be in the gametes, what would be in the pollen, what would be in the egg set. And when they fuse, they produce the diploid complement, which is twice that, obviously. There also exists, obviously, triploid apples, which have 3 times that base number, or even tetraploid apples that can have 4 times that base number. And once you get to that, those levels are now called gigabases. So it's 1,000 million pairs. So this is just getting us to know some of the terminology. Oh, and just to point out how small the apple chromosomes are. Each of those chromosomes is about, because you've got the 10 micrometer scale bar there, is about 1 to 2 micrometers long. So it's 1/1000th of a millimeter long. And it's about 1/5 as long as of the longest human chromosome, and about as long as the smallest human chromosome. So let's see what a nucleotide polymorphism is then. So let me present my DNA now as the well-known double helix, and I've stretched it across the screen. And although we might imagine that genes will represent by far the greatest part of that DNA, they don't indeed. They can represent as little as 2 or 3% of the whole of the sequence. So they're just small islands in long strings of letters that don't really code for anything. So you've got genes and then non-coding regions. And it's the non-coding regions that represent by far the greatest part of the DNA. Now let's look at that and imagine that that sequence is made up of a string of letters A, T, C's, and G's. And this is just a random sequence I made up. But let's assume that is belonging to a particular apple variety. Now let's pay attention to one letter in the middle of that string, the letter A. So in this variety, at that position, we have the letter A. In another variety, we're going to have perhaps a different letter. It could be letter C. We could have letter G, or we could have letter T. So if you've got 4 individuals, you've potentially got 4 different possibilities in that position. They are SNPs, single nucleotide polymorphisms. So it's differences at a single site within the genome. And there are literally thousands and thousands and thousands and hundreds of thousands of these in a genome. And there are more of them in the non-coding region than in the genic regions, because gene regions tend to be conserved by evolution. So now let's bring the two concepts together. Most apples, as I said, are diploid. So if you've got chromosomes in pairs, I've put up 3, again, this is just a mock setup. I've taken 3 diploid apples, Darcy Spice, Adams Pearmain, and Cox Orange Pippin, all diploid apples, I believe. So they'll all have 17 pairs. Look at a single chromosome, and you can see that Each of those has its double helix of DNA. And we can look at a particular position, and you could have both have the same letter there, A, and they would be called homozygotes. In another species, you could have a different combination of letters there, AT, or you could have TT. So A and A are called homozygotes because they've got the same letter in that position. And if you've got different letters, hetero means different, you've got a heterozygote. So let's get back to our experiment. We were asked to see if we could identify the girls, the potential parents of the girls from along Ashton. And we were asked this question by Liz Copas. And here's a picture of it in the field collecting some of the apples for this study, collecting leaf samples for this study. And she's in the field there with Keith Edwards. So what we do, if you look down on the right-hand side, there seems to be a pen there, but that's a little hole borer, which we go into the field. We take a single leaf, we tend to fold it in half, and you take 3 plugs from that. So you get 6 little rings. And it's from those 6 little rings which we're going to extract the DNA. So we don't need a lot of material. Now, traditionally, I put a mortar and pestle up there at the top. When I first started doing science, then I would have done extractions by grinding those leaves up in a mortar and pestle. We tend not to do that. What we do while we're out in the field, we put those leaf samples into a 96 well plate, and there's one there in the background. I don't know if you can I get a pen on there? No, I can't really do that. Oh, I can. So there's your 96 well plate. So I'll just go previous there. And to each of those, you put one particular sample, one particular tree. In the bottom of those, you've got ball bearings, 2 ball bearings. So what you eventually do, you freeze that, put that in the freezer, and then you put it in a big shaking machine, and you shake it vigorously for 2 or 3 minutes, and that simply shatters the leaves, the leaf discs, and releases the DNA. So for this particular study, we genotyped 372 samples representing 245 apple varieties. This included 58 accessions from the Lars breeding program. So some of these were the girls, and the named girls, so the varieties were accepted. Some of them were just varieties that were the product of that, but never got named because they weren't considered of sufficient quality for good cider making. We also added to that many accessions from local orchards and collections. So we had samples from Thatcher's, for instance. And for this study, we used 1,301 SNP markers. So we were looking at 1,300 different sites across the genome for those SNP variants. So in the field, we collect our samples into the 96 well plates. We use the beads to break that up. We add a few solutions to actually release the DNA. We won't go into that. And then we transfer the solutions into a new 96 well plate from where we're going to do the reactions for the SNP analysis. And if we're doing large studies, we can have plates that have 384 wells, or even as many as 1,536 wells. So you do with all this in one experiment. So let's get back to our 3 apples, our 2 homozygotes and our heterozygote. If you were going to piece or amplify across a SNP, for the variety Darcy Spice, variety 1, the only thing we could ever see is an A in that position. If we did that for a sample from Cox Orange Pippin, the only thing we could ever see from that position, which is just a random imaginary position, is a T. If we did it from the heterozygote, Adams Spermain, now, how would we know we'd got these without having to go through all the boredom of actually sequencing it and then reading the sequence to find out it was a... What we can do, and this is getting back to what I said about introducing fluorochromes and making things visible because they emit coloured light. What if we could label anything that got an A in that position so it fluoresced blue, and anything that had a T would fluoresce red, so it could be seen with a camera? Then anything that comes from the heterozygote, from the Adams per main, variety 2, would have both those colours, and those colours would resolve as a yellow or a green. If we had all those in our 96-well plates, any sample that had A and only A would fluoresce blue, and a camera that scanned over the top of that plate would see a blue light. Anything that scanned over the plate with the homozygote for T would see a red light, and anything that spanned over the heterozygote would see a green light. And then you can simply plot that on an axis, and you get separation of the samples, and you can get the computer to automatically call, on the basis of where they fall within that plot, whether they are genotype AA, genotype AT, or TT. Now we've now got to the end of this technical bit. For the sake of doing the analysis of this, it's much easier if you're doing the bioinformatics analysis to move things around as numbers rather than letters. It's easier to compare things. So our SNP calling pipelines collapses those scores into 4 numbers. So AA, for instance, one of the homozygotes, you'd call 0. The heterozygote, you would call 1. And the other homozygote, you will call 2. If the marker failed at that position, you'd give it a score 9. So therefore, again, this is imaginary, you could have SNP profiles. If you had just one SNP, you can only have 3 possibilities. You can be 0, 1, or 2. So that is your profile on the basis of one SNP. You don't just do one SNP. We did 1,300. You did a second. That's what that might look at, a second SNP position. You did a third, and you get that. And I say we did this for 1,200 markers. So you get a profile that looks like that. It's like a numerical barcode. And that is specific for that particular variety. So 211121110 is the barcode for Bramley. On the right, I've just done a little sort of mathematical progression to show you how many different combinations you can actually derive depending on our markers, and that is, you get more than 10 million just for 21 markers, and we're using 1,300. Having done that for your 1,300 markers, what you do is you want to determine a distance for each variety from every other variety in the set. So you're making these pairwise comparisons, and you get, and to do that, all you do is you get the number of calls in common. So what of the 1,300 calls, how many were the same between those two varieties, and how many were different? And you want it so if Queen Cox is being compared with Queen Cox, you want it to be 0. So you take that you take that final number away from 1, and you get this matrix. The matrix is a rectangular matrix, but it's in fact just a triangular matrix. So you can then read it, and the second half of the table is just a replicant of it. So for instance, Queen Cox shares 45% similarity. So 45% of the 1,200 markers will be common between Cox and Lizzie. There were 53% between Cox and Amelia. You can use that distant matrix to either produce a dendrogram, which is something people are probably most familiar with from this sort of work. But for us, it's not all that interesting. It's quite nice to look at, but for our analysis, it was better to use this as a PCO plot. So what you're doing here is of that multidimensional space of all those comparisons, you ask yourself, is there an axis through that data set that explains the vast majority of the variability. And you call that axis 1, your coordinate 1. And then at orthogonal to that, so at right angles to that, you say, what is the next axis that could be pushed through that data set to explain the second most variability in the data set? And then you can plot those. So here on the plot A, on the left-hand side, we've got coordinate 1 against coordinate 2. And the dots on the plot represent the cultivars, as I say, the 350 cultivars that were studied, or a sample that was studied. Those highlighted in green are cultivars from the large breeding program. The other ones, which are just the darks, the black rings, are all the other cultivars that came from other parts. But we're just interested in the green dots because they're from the Long Ashton study. I've also plotted on there the parental species, so the Worcester Pemaine, the Michelin, the Dabinet, and the James Grieve. On the first plot, you can see that the large cultivars are fallen into two main clusters. There's one towards the right and one towards the left. So the first coordinate is separating those samples into two main groups. If we then plot the first coordinate against the fourth coordinate, we got wonderful separation into essentially four basic clusters, with the parents based around those. And we got that some of them lay between Michelin and James Grieve, so we would suggest that they are the girls' products from that particular cross. We get a cluster between Dabinet and James Grieve, so now circled in red, and they would probably be the girls. The parent, the Dabinet and James Grieve would be the parent of those girls, and the same for Dabinet and Worcester Pemaine, and Worcester Pemaine and Michelin. We could show that data in a slightly different way. So rather than it as a cluster, you could just see it numerically. And here, going back to that similarity matrix or the distance matrix I pointed out earlier, these are just figures taken from that. So this is just a small part of that data matrix. And you can see the percentage similarity. So the first group, and it's a similarity. So the greener it is, the more similar those particular varieties are to those two parents. So Angela, Fiona, Gilly, Hastings, Helen's apple, Jane, Jean, Naomi, Sally, they all seem to that have got the greatest similarity, Dabinet and James Grieve. So that's who we believe are the parents of those particular girls. Going further down, Amanda, Betty, Debbie, Lizzie, and it's called Lizzie because of Liz Copas, I believe, and Margaret, I think, is Liz Copas's mother. It would seem that they are the product of James Grieve and Michelin. And also the interesting thing about this is that the Worcester Pemaine didn't seem to be very good, because by far the greatest number of the girls had James Grieves as the male parent. So the best cross was with Jim Grieve. Finally, about this Long Ashton study then, one other thing we did notice is that diploids, and I said the vast majority of apples, about 80, I might be wrong on the figure there, but about 80% of apples, I believe, are diploids. We could actually see from our data set a difference between the heterozygosity of diploids and the heterozygosity of triploids. And I've just put up this little sort of mock-up diagram here. So Dabinet, a good cider apple, I believe, is a heterozygote, and Bramley is a triploid. And this is showing, this is a mock-up suggesting that would be a heterozygote, and that's all that means. So you tend to get more heterozygous SNP markers in triploids than you do in diploids, and I suppose you might expect that. It's got an extra genome, so it's got more chance of carrying a mutation. But the two low and high just represent varieties for which we didn't know whether they were triploids or diploids. And it would seem very much that we could go back into our samples and have a very good guess about, just on the basis of these markers and doing no extra work, because normally you would look for, you'd do flow cytometry or other techniques to determine whether something was diploid or triploid. But this data set is giving a good indication of those things. Anyway, long-Ashton cider apples then. At the end of that work, we were able to publish a paper which was published it's 2 years ago now in Plants, People and Planet. Almost the vast majority of the people had some involvement in Long-Ashton Research Station. So the photograph there is Keith Edwards, who worked at Long-Ashton. You've got Liz Copas and her dog. Liz was one of the pomologists from there. And there's Bob Hughes, and I used to work with Bob Hughes in his greenhouse, because he used to look after my wheat samples. So he was also involved in this research. So anyway, as a result of this, with our 1,200 markers, we were able to identify the parents of the girls, we believe anyway. We also identified some misleading in some samples, but probably that's natural. But coming out of this work, we asked ourselves the questions, could SNP markers efficiently replace SSRs? Well, SSRs are microsatellites, single sequence repeats for routine apple identification. So normally, I think if the standard procedure for genotyping apple at the moment is to use SSRs or microsatellites, which I could explain afterwards, but I'm not going to explain here what that precisely is. They use, I think, 12 of those, but they're slightly harder to automate. So we asked the question, could we replace those with our SNPs and get a minimum number of SNPs that would allow you to accurately identify quickly your varieties? So this is our concept of minimal markers. How many SNPs are required just to identify a variety? We want this to be rapid, we want it to be accurate, and we want it to be cheap. Now, given the last year we've had here, we've used this minimal marker concept for apples, and I'm going to talk about that now. We've also tried to apply it to wheat, but because of last year, someone in our lab suggested, why don't we try it on COVID and look at the virus? So we've also tried it on that. So what is this minimal marker concept? The minimum marker concept is this. For each individual SNP marker, and we had 1,301 markers, so for each marker you create a matrix, and the matrix is of the varieties, and you're just simply, is marker 1, for instance, able to distinguish between variety A and variety B? D means yes, it distinguishes them. S means no. So marker 1 could distinguish A from B, but not A from S. It could distinguish A from D, but not A from E. So that first marker for these 7 varieties was able to distinguish 10 of them. So it had a score of 10. Marker 2 was able to distinguish 11. Marker 1,300 was able to distinguish 9. So you create this matrix for each of the markers, and then you go in and say to yourself, and this is all done with software written by people within the lab, not by me, but by Gary Barker. Say, okay, tell me which of those gives the highest score. And in this case, it's marker 2, because it gives a score 11. And then you select that and say, okay, I'm going to select that and keep that. And then when I run the process again, how many of the remaining markers, so which of the remaining markers gives me the best ability to separate my samples? So you have a second iteration. If you look at this second iteration, even though the score of 1 was higher for marker 1 in the first round, it's not now. Marker 1 only adds an extra score of 2. So it would only add the ability to distinguish an additional 2 varieties, where marker 1300 allows you to distinguish an extra 7. So you then would select that marker and add that to your minimal marker set. And the script iterates through that process, adding additional markers until no further resolution is arrived at. Or, and this is what we really want, all varieties have been resolved. So this is all just done with a piece of software. We're not talking about biology at the moment, we're talking about bioinformatics. Okay, so we ran this on the data set, the 350 varieties with 1,300 markers to find a minimal marker set. And we came up with 21 markers able to distinguish all the cultivars in that study. And at the bottom, there is the Bramley, Cox Orange Pippin and Worcester Romaine. They are the genuine SNP profiles for those 3 varieties. We took those minimal markers and we wanted to test them. We want to say, well, are these actually very good? Can we use these and will they really work in the real world? So we approached people at Brogdale and said, can we come to your collection? We know nothing about apples. We don't really understand them very well at all. It's not like I can recognize something in the field very well. But can we come as a blind test, take samples of all the trees in your collection, and we'll run our markers against them, and then we'll tell you what we think you've got, and you tell us if we're right. So the experiment design then was to use 21 of these markers on minimal marker set. The number of samples we had were essentially 2,500, so 2,482. And the samples, most of them came from the National Fruit Collection at Brogdale. We had some samples from the Welch Botanic Gardens, and we included all the samples from the Long Ashton study as well. Now, based on name, and this is really important, based on name alone, 2,123 distinct varieties were in that collection. 1,919 of these were singletons with just got one sample, where 204 had at least one rep. And as an example is the variety Yeovil Sarah, we had 24 technical reps. So what we'd done is we'd extracted DNA from one leaf of one tree and we divided it 24 ways, just to see that, to check whether our technique was reproducible and what level of error is likely to occur, because obviously all techniques have error. What are our expectations then? Our expectations are that we should be able to distinguish all the singletons. They should each have a separate individual SNP profile, which is unique to them. We should also be able to unite all the replicates. So we don't know which they are because it was a completely blind test, but we should find things that the SNP profile should be repeated. And then we'll put those together and say, yes, they've got the same name. So we've united the replicates. So for the singletons, we're expecting 1,919 unique profiles. And for the 204 profiles, and for the replicate samples, we'd expect 204 profiles that are shared by more than one sample. So in total, we're expecting to see 2,123 different profiles in our set. And what disappointment. I was doing this work at the beginning of last year, so we just entered lockdown, and I said, look, I'll do this, I'll do the analysis and I'll write the paper, and it'll give me something to do while I'm sort of just sitting alone at home, which is not true because my wife also works from home. But so my first thing was disappointment, because we're expecting to see, as I've just said, these 2,123 profiles. What was our result? Rather than obtaining 1,919 unique profiles, we obtained only 1,495. And rather than having 204 profiles that were shared, so finding where the replicates are in the samples, we had as many as 325. So our total profiles was 1,820, and we were expecting it to be 2,123. So let's have a look. Why did we fail? Where did it go wrong? So here's some of the things we had in the collection. From the Welsh collection, we had something called Hafal Enri, and my pronunciation is probably very poor. 2 of those samples, and we had something called Bardsley 1 and 2. Now, different names, to my mind, they should have different profiles. Certainly don't. And you can see for marker 8, there was a fail in there. But the program that I've written to pick those out ignores the fact that there's a missing data set. From Brogdale, we had 3 samples of something called strawberry pippin, and each of them had a different profile. We had 4 samples of Jennet Moyle, 2 from Brogdale, 1 from Shepton Mallet, and 1 from Thatcher's. One of the samples from Brogdale was completely different from the other 3. So it was rather disappointing. It looked like we'd not really had much success. But for the variety Yeovil sour, for which we had 24 samples, we had perfect reproducible profiles. And then marker 11, we've got 1 that's failed, but 1 out of all those markers is actually very low fail rate. To tell the truth, you can see there's only 23 samples. The 24th sample, for whatever reason, failed, and it might be to do with the position on the plate. Certain samples that lie to the edge of a 96 well plate, sometimes when you have to put over almost like a cling film that stops evaporation when you're doing the processes, and things at the edge of a plate occasionally dehydrate and dry up, and then their samples fail. And that might have happened to the Yeovil Sour 24, because basically none of the markers worked. But anyway, so for that particular sample, it looks like they had working. Let's get back to the Bardsey Island apple, or this apple Henji, however you pronounce it, which comes from the Isle Henji, which, if you don't know, is a very small island off the tip of North Wales. And it's that, can I do that again? Yes, I'll bring up that. It's that little island there. Well, some people claim that this is the rarest apple in the world. It was found on this little island, 1 tree, and there's a picture of it growing along the side of a house. This is a very windy island, so the tree itself actually gets pruned by the salt wind that comes past, so it hugs the house and goes up over the ceiling, and it never really extends out, because if it did, it would be killed by the salt wind that's there. So it's well protected where it is. But anyway, in 1998, somebody who knew something about apples identified this. Well, that's an unusual apple. It could be something that's quite distinct and rare, and they took it back to Brogdale and they identified it as a distinct variety of apple. But anyway, as far as I'm concerned, that is good news, because in fact the English call that island Bardsey Island. So those 2 names, which for me should have had completely different SNP profiles, were in fact the same, a very same apple, one with its Welsh name, one with its English name. And in the end, I went back to people at Brogdale and say, look, in part it's worked, and we've got some nice results, but we've still got a lot of problems. It doesn't make a lot of sense. And people at Brogdale, Matt, said to me, well, you could look at the MUNQ codes of these samples. Now, I didn't know what MUNQ codes were, and he told me that these are Malus unique codes, and these have been assigned on the basis of this. This is from Brogdale again. This is from the National Fruit Collection. These have been assigned over the last 10 years based on phenotype, but also genotype, based on these single SSR markers, which are the microsatellite markers, and DART markers. So 2 of the marker systems. So they've been assigned numeric codes that say how faithful that those particular varieties are. What we discovered then, when I went back to my data set with these new MUNQ codes superimposed on the National Fruit Collection data set, I found out that different names could have the same MUNQ code. So apple varieties Brabant Bellefleur, de Flandre, Franc de Pommier, Héroux, they are all MUNQ code 1. So they're just synonyms. We've got the Bard's Island apple at the bottom, Bismarck made of Kent, synonyms. Melacarla, Rosa di Caldro, synonyms. And then on the other, we've got all the different synonyms for various golden delicious type things. We also discovered that some things have the same name, but different MUNQ codes. Now, this is part of the process of the National Fruit Collection, the people at Brogdale getting their collection sorted out. So genotyping exactly what they need. So they've done it using these SNP markers and dark markers. And they recognize within their collection that they have 3 things called strawberry pippin, but they're in fact 3 different varieties with very different MUNQ codes. The 2 genet moils, the genuine genet moil is the 827. So the people at Shepton Mallet, the people at Thatcher's have got a genuine genet moil. One of the samples at Brogdale is not a genet moil. So in the end then, although we thought we'd failed, we hadn't. We got something like 99% agreement between our study and what the people at Brogdale saying the samples were. But we didn't get 100%. Why not? And the reason is because, as I say, errors occur, mistakes happen. We were going into that field not as experts on apples, and well, this is just showing that this isn't necessarily an error, but I'm just showing the sort of things that can happen. And obviously you are more aware of this than I. Here you've got rootstocks and the scion are both flowering, and this is 2 different trees. So if I'd gone along there and wasn't paying attention to the leaf sample I was collecting, I could have been collecting a leaf sample from the rootstock, thinking it and naming it what the scion was. So that could be an error there. Things get misnamed. When we get back to the laboratory, we were dealing with 2,500 samples. We could have mislabeled something. You could have dropped something into the wrong well. And also some of the things that went wrong, I noticed was when I went back, Matt at the garden said, you know, I've got this misnamed, I've named it this. He said, yeah, that's strange because those 2 apples lie together in the orchard. So obviously I'd made a mistake during collection. So that's why we probably didn't get 100% accuracy. Now, I've got a PDF I want to look at over there. Is that going to take me somewhere? Let me get rid of that. And I was hoping that was going to. No, it's not taking me to where I wanted to go. Never mind. So we can say, how do you show the data? Now, this is a post I've got just behind me. I've got it rolled up. In order to make a data set like this visible to people, you need a very, very big poster. And I made a poster which is, I think it was 2 meters by 80 centimeters, a huge poster. And this is just a representation of it. In order to read that, you need to go. You'd need to have a poster of that big size. Now, I wanted to go to the interactive. Want to keep this call. Just a minute. Okay, so I'm going to say there's that huge poster that I've got curled up behind me. a better way to present this, I actually introduced this data onto the interactive tree of life. So if you go to this site that is referred to in the paper we published on this, you can see this tree in a circular form, and there it is. And the good thing about this is you can actually project things onto it. So I could click over here and I could get it to show me the ploidy of the samples within that collection. I could get it to show me the level of heterozygosity. So the darker the color, the more heterozygous. And you can see that heterozygosity corresponds to some extent, quite a good extent, with ploidy. I could also highlight conflicts between our data set and what MUNQs, and there's a very small percentage. But the good thing about this is you can actually search it by name. So if I put, I don't know, let's put Dabinet in there, and then I can see where that is, and you can see what it does is it takes you to that part of the tree, and I'll try and move it over. I need to get it a bit smaller now. Anyway, it's rather cumbersome because the data set is so large. There's 2,500 samples in there. Let me just do another thing. I'll just click on a subset of the tree. Add to Poon tree, and then you can do that. Take those off, take that off, and here you can see that you can get subparts of that tree, and here you can see that we've got some of the girls together: Jilly, Angela, Willie, not really a girl's name, but it's one of the girls, Jane, 3 counties, Hayden, Tracy, Tina, and you can see that they cluster together within the tree. So let me come out of that. Let's take this back over here, display settings, swap things. Okay. And as I say, we published an article in that in +1 at the end of last year. Just briefly, I'm coming towards the end now. Just briefly then, as I said, the very same concept you could use with variants, so variants of concern. And we developed a panel to look at this. We pulled out the Wuhan sequence, reference sequence of the SARS-CoV-2, and there's a sample of its SNP profile, and you can see many samples. If there's just a star here, that means those samples are exactly the same as the Wuhan for that particular thing. But we were able to identify some of the samples, the variants of concern. So and we've put this forward to the Public Health England to have a look at to see if it's something they could use for rapid analysis of variants of concern. Anyway, and we published something on that last year. So finally, let's get to the end of this. The apple hunt. Again, this was a lockdown project to some extent. We'd sent this out just before the lockdown and asked people to send in their apple samples and not to tell us the name. So what we were going to do, we wanted them to send in apple samples with code names for their apples. And we've got wonderful letters set in, and even people sent in photographs of their apples. So this is a very interesting sort of red looking apple that someone's got in their garden. But anyway, for each of the apples, we wanted to give a code name. So we could then genotype them. And then once we'd done the work, the people would, and I created a new website for this, people would come onto our website, and it's called Apple Source. I'll show you in a minute. They put in their code name for their apple, and they find out what their apple is. We received 824 samples, each with their secret code. And we had piles and piles of letters in the freezer until we could do the work. We did the extraction, as I talked about earlier, and we ran the analysis.
Hi there, Mark. Sorry, I'm just going to interrupt a moment. We can't actually see your screen. It's stuck on the on your poster.
Oh, let me. So Let me see if I'm going to come out and come back in. Just a minute. Stop sharing. Right then.
That's right.
Yeah, it was me trying to be clever and actually jump out and jump back in. Never try and be clever. Let's allow. So if I run that.
We've just got a couple more minutes of the
yeah, well, I'm really. Can you see now?
No.
You can't see now. Why can't you see now? What if it says you're showing another application window. If I put that over there, can you see that?
No, at the moment it doesn't look like you're sharing, unless I can't just see it. But I think no, nothing being shared.
Oh, well, that's a shame. But let me just finish off. It's just 2 minutes anyway. You can't see it. So just to finish off, so we sent off these samples, but people sent in the samples. They sent in their codes when we've done the analysis. They simply put the codes into our website, and it told you the varieties that they'd sent in. And it's a shame I can't send it to show you that final slide, because it's showing you the sort of samples that they'd got. But most people, the top hit of all the sort of unknown samples that sent to us were Bramley apples. The next was Cox Orange Pippin. Then we got to Bulmer's Norman, then Blenheim Orange, Lady Landburn, as they were the top ones in the collection. Anyway, then to finish, that was last year's apple hunt. This year, we're wanting to repeat this experiment. We have a new student we've taken on specifically to do that work, and we want to go out and sample apples in as many of the local orchards that we can. And to some extent is for looking for apples that people really don't know the name of. So we can find if there's anything interesting out in some of these smaller orchards. I think we've already agreed with Thatcher's to do this for them and one or two other gardens, but we're really keen to get people to contact us and let us come, because we'll come out later in the summer and collect leaf samples from your trees, so then we can identify them. If we've already got that sample in the data set because it was present in Brogdale's, we'll be able to give it a real name. If not, we'll be able to determine whether it is something distinctly different and new. So I'm sorry about the last few slides, but thank you for giving me the opportunity to talk. If you've got any questions, please ask away.
That was brilliant. Excuse me. That was brilliant. Thank you ever so much. It took me back to my university days, part of that session as a biochemist, which was a real revision session for me. So that was really interesting. A couple of very quick questions. Mike Brown asked, can you give us the link for the interactive Tree of Life? That's a bit that we couldn't actually see.
Yeah, I will give you that. I'll write it down in here in a minute. I need to go and look at it myself. I haven't got it on the top of my head.
That's all right. There was a suggestion given by Gillian in the chat, but if you could just have a look at that, or type in the chat when you find it, or even in the main event chat after the session would be great.
Yeah.
Jackie Denman, who I was going to mention anyway, has just put a question in. What do you consider local? Sorry, the questions are really coming in there. What do you consider local when you're talking about local orchards for the apple hunt? Well, I know that you're looking at our area here in Herefordshire.
Well, I suppose to us it's local. It's almost anything to the southwest of us, but Gloucestershire. I mean, I know that's not but the southwest. But I don't think we'd turn anybody away really if they'd got an interesting collection. We want to do this study. We think it'll be useful, and it's also very interesting to us. I mean, this morning I had a quick conversation with somebody suggesting that there's somebody out in Kyrgyzstan at the moment collecting Cerevisiae, and it probably would be quite nice to get some leaf samples of those to look at as well, just as a sort of as an out.
Absolutely,
sample.
absolutely.
Yeah,
so Mike Brown is your man there.
That was it. Mike Brown was the name.
Yeah. So he's asking the questions. Are the samples from the UK? So I think we've answered that one. Darlene Hayes. Hi, Darlene. It might not be convenient for you to come to my tree, but is it possible to send you leaves? She's in California.
Well, I'd love to come. You probably could. Yes, you want them to get here fairly quickly. I mean, one of the problems with the hunt last year is that simply some people sent us whole branches in, which was fine, but we had 99 times more than we needed. We just needed one leaf essentially, but we need that to arrive in a decent condition. If they're around sort of the rotting, they've been too wet when you put them in the thing, it becomes difficult to externate. Yes, but yes, as long as you can get to us fairly quickly, we can do it.
There's a couple more questions. I've got another session now to present, so I'm going to have to leave it there. Mark, thank you so much for giving us all that time in that presentation. Really, really enjoyed seeing all that. I hope you can stick around for the rest of the conference. And please, if anyone's got any questions for you, if you're around, if they can pester you by direct message.
Well, they can. I mean, if you've got my email, which is just mark.winfield@bristol.ac.uk. Just send something to me or through the chat. I'm happy to respond to and certainly get in contact if you've got an orchard and you want us to look at your samples.
Brilliant. I should imagine you're going to be inundated.
Good.
Thanks ever so much, Mark.
No problem.
Bye-bye now. Thanks everyone for watching. Bye-bye.